actually trained as a cell biologist, molecular biologist at the Harvard Medical School. So soon after I finished my postdoc training, I actually went to work for a company, the iTech pharmaceutical company. We were the first one actually developed the first anti-VEGF drug for macular degeneration. And after that company, I went to do some cell-based therapeutic research in a smaller company, a startup company also in Boston. After that, I was lucky enough to actually return to academia and work in University College London, Institute of Ophthalmology, looking at various ways to protect the retina using novel neuroprotection method, including using VEGF, and also try to develop new target for neovascular AMD. And in 2013, I was recruited, again, very lucky, again, back to Scape and Lab Research, where I actually got my graduate degree to work with Professor Patricia DeMore. And I'm just starting my new lab right now. And my lab, because I have a bit of background in using anti-VEGF drugs to treat neovascular AMD, I know how that drug works. I think it's a wonderful drug, but it has its limitation. I understand its limitation well. And I think after 10 years of using that therapy, it's about time we think about moving on and getting a next generation therapeutic, if you will, that either make the existing anti-VEGF drugs better, or maybe even finding a new way to treat the disease that may be slightly more effective, less toxic, for example, because even the anti-VEGF drug nowadays, we have a lot of people don't responding to it. And in the long run, you will look into 5, 6, 7 years, the vision improvement do not retain, and there's still degeneration going on. So that is the kind of area that I would like to look into. So in terms of my lab, like I said, I'm just starting now, building it up. So there are a couple major areas that I want to look into. One, like I mentioned before, anti-VEGF drug is good, but it doesn't really treat the fundamental problem. For example, it only suppresses vascular leakiness. It would be wonderful if we can find a drug or some kind of therapy that would actually stop the CNV from developing from to begin with, so you don't have to worry about the symptom afterward. So I'm looking at that area, looking at pathway that actually responsible for the pathogenesis of CNV. For that area, we have some novel target we are looking into, including, I can tell you, for example, pathway that involve the innate immune response pathway, for example, the toll-like receptor, or the CCL receptor, and so on. And we have some interesting data. Hopefully, we'll be able to publish that soon, and so on. And the second area is very dear to me, which is neuroprotection. Because when I was in iTech Day, we actually was looking into how we can protect the retina from degenerating on top of suppressing the vascular pathology. So that area is slightly more challenging, if you will, because so far when we try various ways to protect the retina from degenerating, we seem to have mixed data, to put it lightly. So that's the area I would like to explore more in the near future by, for example, looking into the mechanism behind, for example, VEGF, which turned out to be a very, very potent neuroprotection. And at the moment, I know everybody is thinking about that as the bad molecule, but in fact, from a developmental point of view, it's actually a wonderful molecule. So I would like to harness some of the good sides of VEGF, for example, neuroprotection side of it, but delete the bad side of it by engineer some kind of VEGF molecule that we can use, for example, for neuroprotection. So that's ongoing research, the two of the major area.
Well, that's obviously a very novel approach because people either characterize VEGF as bad or potentially good. And what you're saying is that there are elements of VEGF that are beneficial.
Absolutely. So this is, if you look back to the literature, the discovery of VEGF in the knockout and so on, we actually know this molecule is very important from the beginning. So something so fundamentally important, probably won't be all bad. Everything just like, you have a good side, a bad side. Too much of it is going to be bad, not enough, for example. So, but we have data in the past 10 years suggesting that the VEGF molecule you can divide into different domains, and there's some certain domain that actually is associated with the bad effect. For example, increasing inflammation, which is the heparin binding domain we know of, and but not the rest of the molecule. So it's really complex molecule and signaling. So we need some time to dissect out the pathway, and maybe try to find a way to, like I said before, find a way to enhance the good size of it and engineer the molecule in a way so you can find a way maybe use it for the beneficial effect, but do not have the baggage of the side effect for causing leakiness, for example, or causing angiogenesis when you don't want it to. So.
Because we heard about, for example, the increase in geographic atrophy with long-term use of VEGF inhibitors. And clearly that makes physiological sense. And if you are reducing circulation, you're reducing perfusion, then geographic atrophy would increase. So are you looking at that element in terms of improving VEGF inhibitors going forward? That they might not be that intensely inhibiting so that they don't reduce the circulation to the point that they accelerate geographic atrophy?
I think the GA data, I'm sure you're referring to the CAT trial, the data from the CAT trial, the increasing use of the avastin in particular that causes. I think there may be two elements to it. That one is the direct blood flow effect that you mentioned. The second might be, in my opinion, it could be a direct toxicity effect of the anti-VEGF therapy to the neuronal cells. So we have data, myself, I was involved before, suggesting there's a direct toxicity effect anti-VEGF for retinal ganglion cell. And of course, Professor Demore, Margali, also escaped in our research, also show anti-VEGF could have a devastating effect to the photoreceptor. So all these things combined, suggesting there is a potential toxicity, direct toxicity effect to the neuron. So there's twofold to that. And yes, you're right. So when I think about anti-VEGF, I think it's not going to go away because it's so effective. When you think about it driving it up, so the leakiness so nicely and then restore the vision so drastically, I think we should keep it around. But the usage, for example, maybe the maintenance therapy we can think about in the long run. So for example, the ideal situation would be if we develop another way to suppress the CNV from growing. That's what the novel molecule I'm talking about. Use the anti-VEGF as a first-line therapy for the first year. Dry up the lesion, and then use something less toxic for maintenance therapy for a long time that could work. And conceptually, that's actually maybe better than just keep using the anti-VEGF because people develop resistance and so on as well. So that will be one area I think we can think about as well. I can tell you experimentally, we really need to demonstrate, for example, the neuroprotection idea. Number one, we need to demonstrate I can actually engineer VEGF that protect but do not have the bad effect. So I'm working on that. That's the immediate goal. If we can actually do that, some proof of concept experiment, either in cell-based experiment or animal experiment, that'll be, very good, and we can open up a lot of more window opportunity after that. And if that work, who knows where they would lead us, right? In the other area, when I was talking about novel pathway that actually involved in the pathogenesis of CNV, again, I have great target, and I need to do some proof of concept experiment to show the functional role of those target in the pathogenesis. That means if I was able to suppress not only the leakiness, but also the CNV not being developed altogether in certain animal model, that will be the milestone, and then I can move on to really thinking about how to target a pathway for translational applications. So.